Weird that like next week is I've like lost motivation and I need to keep going because next week is like the last lesson, which is good.
Is our final next week?
Mm-mm. We don't have like a lesson due on the week of the final, thank goodness.
Oh, I don't know. I'm just done.
Me too. Let's see. Okay.
Have I looked at the key? I think I did.
I don't think I did.
I haven't looked at the Applying Genetics key because it wouldn't pop up.
Oh, really?
So my answers are probably like not right.
Yeah, I haven't done the Applying Genetics yet. I need to get that, working on that. Okay. I'm gonna pull this up. All right. So number 1, what 2 regulatory proteins are involved in the lac operon? So that's the lac repressor and then the catabolite activator protein, which is CAP.
Mm-hmm.
Now, the 2 effector molecules in the lac operon are allolactose, which will bind to the lac repressor and it will release it from the operator, and then cyclic AMP. And so that will bind to CAP, allowing CAP to bind to the activator binding site. For C, what 4 DNA sequences are involved in regulating the transcription of the lac operon? So that's the operon, that's the CAP site. And then the lac promoter, and the lac operator, and then the lac promoter. And then the 3 structural genes that are found within the lac operon are lac Z, lac Y, and lac A.
Yep.
Okay. So suppose we isolate an E. coli strain that produces an inactive lac repressor protein that cannot bind to the operator sequence of the lac operon. What happens to the expression of the lac operon genes? So it says an E. coli strain called lac L negative produces an inactive lac repressor protein that fails to bind to the lac O sequence. This lac L minus strain displays constitutive expression of the lac operon. The lac operon is transcribed both in the absence and in the presence of lactose. And then is this advantageous or disadvantageous to the cell? And why it says constitutive expression of the lac operon is disadvantageous to the cell. If the lac operon is transcribed constitutively, then the enzymes beta galactose and lactose permease and galactoside transactylase are produced even when lactose is absent. Translation is a very expensive process in terms of cellular energy. By producing these enzymes, even in the absence of lactose, the cell wastes 1 ATP per amino acid, 2 GTPs per amino acid, and the final GPT to terminate translation for each copy of these 3 enzymes. That's good. Okay. Do you want me to just keep going?
Yeah, you can just keep going.
Okay. In the lac operon, how would gene expression be affected? So in parentheses, inducible, non-inducible, or constitutive if one of the following DNA sequences were defective. So if the lac promoter were defective, a mutation in the lac promoter would make the lac operon non-inducible. In the lac promoter mutant strain, sigma factor and RNA polymerase cannot bind to promoter under any condition. If the operator site had a mutation, that would make it constitutive. The lac repressor cannot bind to the lac O under any condition in that strain. Therefore, the sigma factor and RNA polymerase can bind to the lac P promoter either when lactose is absent or when it's present. With the lac Y gene, and that would make it non-inducible. Lac Y is the gene responsible for making lactose permease, the enzyme in the outer membrane of the E. coli that is responsible for moving lactose from the environment to the cytoplasm of the cell. Even when the lac operon is repressed, the lac operon is leaky, producing a few copies of the lactose permease per cell. However, in an E. coli strain with a defective LAC-Y gene, no functional lactose permease will be present in the membrane, so no lactose will ever be moved into the cell to be converted into allolactose. And then with LAC-Z gene, a mutation in the LAC-Z gene would make the lac operon non-inducible. LAC-Z is the gene responsible for making beta-galactosidase, the enzyme that converts lactose to allolactose. Even when the lac operon is repressed, the lac operon is leaky, producing a few copies of the beta-galactosidase per cell. However, an E. coli strain with a defective LAC-Z gene, no functional beta-galactosidase will be present, so lactose will never be converted into allolactase.
Did you say A was inducible?
A is non-inducible.
Okay.
Yeah. Okay. All right, so the number 4, this question refers to, I guess they would say Jacob, Monod, and Party experiment. I remember like learning about this in other classes. I've actually already taken molecular biology. I graduated from BYU in Provo like a year and a half ago. I just needed the genetics portion, so it's kind of funny because all this stuff I've already kind of learned. But why was beta-O-nitrophenyl galactoside used in the experiment? So that is a substrate for beta-galactoside. When the lac operon is transcribed, beta-galactosidase is produced, and beta-ONPG is converted into its product ONP, which is yellow. As a result, beta-ONPG was used in this experiment as a convenient means to determine if the lac operon is transcribed when a yellow product is observed and the lac operon is on. Why was no yellow observed in one of the 4 tubes? No yellow was observed in the reaction in which the LAC-L plus and the LAC-L minus merozygote strain was incubated in the absence of lactose. In the LAC-L plus, LAC-L minus merozygote strain, the LAC-L plus gene on the F prime plasmid produces lac repressor proteins that binds to both copies of LAC-O plus in the cell in the absence of lactose. When the lac repressor is bound to both operators, both copies of the lac operon are repressed. What is another method that could be used to measure the level of expression of the lac operon? So that could be, one could attempt to measure the levels of the lactose permease, the pump that moves lactose from the environment to the cytoplasm of the cell. For example, lactose permease levels could be determined by measuring the movement of the radioactive lactose into the cell. If the lac operon is transcribed, radioactive lactose would move into the cell. Alternatively, antibodies could be used to detect the presence of any of the 3 lac operon enzymes in Western blot. Why was the optical density twice as high in tube 4 as in tubes 1 or 2? Tube 4 contains the lac plus / LAC-L minus merozygote strain incubated in the presence of lactose. In this environment, the lac repressors will be released from both copies of the LAC-O, and transcription of both copies of the lac operon would occur. Since there are 2 copies of the lac operon in the cell, the yellow color produced upon lac operon induction will be twice as intense as observed in tubes 1 and 2. Okay. Man, these are like really long answers.
Yeah, I think she just expounds on them because like most of
Understand.
yeah, it made it make sense if she just gave us the answer, so she started giving us extra.
Yeah, which is good.
Mm-hmm.
Okay, so describe how the lac operon is regulated under the environmental conditions listed in parts A through D below. Indicate why it's transcribed or not transcribed. Yeah. So no glucose or lactose present. Lac operon will not be transcribed in the absence of glucose and lactose. In the absence of glucose, CAMP levels will be high. CAMP will bind to CAP, and the CAMP complex will bind to the CAP site on the DNA promoting lac operon transcription. However, in the absence of lactose, no allolactose will be formed, and the lac repressor will remain bound to LAC-O. When the lac repressor is bound to LAC-O, the lac operon is not transcribed. All right. Glucose present or no lactose. The lac operon will not be transcribed in the presence of glucose and in the absence of lactose. When glucose is present, CAMP levels are low. Without CAMP, CAP will bind to the CAP site on the DNA and transcription of the LAC operon will not occur. In the absence of lactose, no allolactose will be formed and the LAC repressor will remain bound to LAC-O. When the LAC repressor is bound to LAC-O, the LAC operon is not transcribed. If lactose is present but there's no glucose, the LAC operon will be transcribed when the lactose is present and there is no glucose present. In the absence of glucose, CAMP levels will be high. CAMP will bind to CAP. CAP complex will bind to CAP site on DNA, promoting LAC operon transcription. When lactose is present, lactose is converted to allolactose, and the allolactose binds to LAC repressor. The LAC repressor is then released from LAC-O, promoting LAC operon transcription. And D, glucose and lactose present. LAC operon will not be transcribed when both glucose and lactose are present. When glucose is present, CAMP levels are low. With CAMP, CAP will not bind to the CAP site on the DNA, and transcription in LAC operon will not occur. When lactose is present, lactose is converted to allolactose, and allolactose binds to LAC repressor. The LAC repressor is then released from the LAC-O, promoting LAC operon transcription. However, since there are two go signals are required for LAC operon transcription, the system remains in an off state. So that's all. I don't think this one was too bad. What do you think?
No, I didn't think this was too bad. The only thing I didn't understand was the plant genetics, like part 5 of it.
Yeah, I haven't done that.
Actually, and 4. 4 was kind of like shaky, but I can't open the key review for some reason. Like it's jumped from like the quiz to the key review from like the group work stuff. So I don't know what happened, but my iLearn is sketchy.
Yeah, that's what it sounds like.
I don't know. So I just don't know the answers yet to the other homework, but I'll figure it out eventually.
Yeah. So did you turn in 13 and 12?
Yeah, so I turned 12 in just today and this morning, and then I turned in 13 on Wednesday. Because I thought, I don't know what I was thinking, and I was like, that is so weird. She put 12 like as 13, but then I realized today when I opened up 12, I was like, wait, there is a 12. So I felt really stupid, but it's okay.
Yeah, at least you're ahead, right? Awesome. All right. Well, have fun on your little cruise thingy.
Thanks.
See ya.
All right. See ya.
Do you want me to send it to you or do you want me to just post it?
You can just post it because I still don't have access to that group thing.
Okay. I'll just post it then.
Thank you.
Yep. Bye.